So my lab is largely interested in creating and examining DNA variation in both living Native American individuals as well as their ancestors through paleogenomics to infer the evolutionary history of Native Americans, starting from the initial peopling of the Americas all the way through to the effects of European contact and colonization. And so after European contact and colonization, the environment in the Americas, as well as Native American genomes, changed dramatically, as well as the diversity, as infectious diseases such as smallpox and other diseases, as well as warfare, caused Native American populations to decline rapidly. And so that's something that we have to keep in mind when we're using DNA variation to infer population history in the Americas, is we have to know that what's the diversity represented today may not be consistent with what it was pre-European contact. And so today I'm largely going to focus on my work with living individuals in the Americas, and I'm going to leave a lot of the ancient DNA or paleogenomics that has been done in the Americas to Dr. Eski Willerslev, who will talk a lot about the cutting-edge research tomorrow. So here is a map of living populations that have been sampled for genome-wide data in the Americas, generated by David Reich and Andres Ruiz Linares and many other co-authors. And what you can see is that there are large gaps in the sampling. For example, the United States doesn't have any sampling at all of Native American individuals or communities that contribute to the data set. And so these large gaps limit what we can say using our genetic models. We can probably give broad scope types of information about the peopling of the Americas, but we can't do any of the regional analyses where you would need much denser sampling in different regions of the Americas to say anything meaningful. So how can we actually overcome these hurdles of having these large gaps? Well, a lot of these gaps, especially in the United States, as a result of social opposition to sampling, largely in part due to the transgressions of past and current researchers with Native American communities. And so there are, I believe, ways forward by having researchers today actually collaborate, creating trusting, mutually beneficial relationships with these communities to address questions that they are interested in, as well as questions that we, the researchers, are interested in. And so we can do things like after the papers are published and the analyses are, well, even before that, after results are generated, going back to the communities to discuss with them the genetic patterns that we're seeing to get their insights into what may be going on. We can also do things like have them work with us to work with other communities to help develop a broader data set, right? The other thing that we can do is something that I've been involved with for the past few years. Oh, sorry. One last thing is we recently published another data set in PLOS Genetics last month that increases the sampling, but still only within British Columbia. So there are still these large gaps that are out there. So one other thing that we can do to help potentially increase this sampling and get Native American individuals involved in research and genomic research is to train the next generation of Native American students in the sciences to become genomicists and to have interests in doing this type of research to address questions that they're interested in and their communities are interested in, and they can devise experimental designs or study designs that are useful and diverse, right, relative to collaborating with an outsider who may not approach the questions or the study design in the same way. And so to help with this, we created the Summer Internship for Native Americans in Genomics, and this is a one-week workshop that takes place over the summer. Usually it's at the University of Illinois at the Institute for Genomic Biology. This last year we did it at the University of Texas in Austin, and we usually have between 12 and 14 Native American students from throughout the US and Canada and Mexico, as well as Hawaii, Native Hawaiians, come for a one-week workshop where we train them in some molecular biology techniques and show them how to extract DNA, as well as do some amplifications or some DNA sequencing. And then there's also a bioinformatics component where they download large genome-wide data sets and they perform different analyses and recreate analyses and figures from leading papers in Science and Nature. That is very encouraging for lots of these students. And then we have a portion that's on the ethical, legal, and social implications of genomic research with indigenous communities. And this is often a very heated time of the workshop where we discuss past transgressions as well as current positive collaborations that are going on right now between non-indigenous researchers and indigenous communities. And so I think by having Native American students become genomicists and answering questions in their way, we're going to create diversity in the way studies are designed that will give us insight that we don't have right now. Okay, let's move on. All right, so now I'm going to move on to talk about the Beringian standstill theory, or the Beringian incubation model. So this is a study based on mitochondrial genomes from living individuals. There's no ancient DNA involved in this study. And so in this study, what we did, this was published in 2007, is we sampled thousands of individuals from Northeast Asia, from over 24 populations. And then we sampled over 600 individuals from communities in the Americas. And we sequenced their complete mitochondrial genome and identified the diagnostic substitutions or mutations, and then we screened all of these individuals for these diagnostic substitutions or mutations. And we found a few interesting patterns. One of the patterns that we found is that there were mitogenomes that were present in high frequency in the Americas that were absent from Northeast Asia. And this is very surprising because usually you can find what's called a private allele or a genetic variant that's unique to a region or an area, but it's usually in very low frequency. In this case, we're finding these genetic variants in extremely high frequency. And this also fit with other genetic patterns that we were seeing in Native American populations in the autosome as well as the Y chromosome. And so we took this information and from the mitochondrial genomes, we used the molecular clock to get a time of the most recent common ancestry. And it was just after the last glacial maximum, around 16,000 years. And so we took that information along with the archaeological and paleoclimatological information, and we developed this model. That's the Beringian incubation model, but seems to be known in the literature as the Beringian standstill theory. And so this model goes as follows. There was an initial peopling of Beringia, and then during the last glacial maximum, as Ted discussed, there was perhaps a group of individuals that stayed in Beringia in a refugium, and then at the end of the last glacial maximum, during that last glacial maximum, these individuals in Beringia were isolated from other populations in Northeast Asia. And so during this isolation, they were actually able to evolve new genetic variants that gave them derived mitochondrial genomes that when they spread into the Americas, caused them to be unique mitochondrial genomes, where the closest related mitochondrial genomes were in Northeast Asia. And so this is a model, a hypothesis, and so there are other patterns here that we can see. So when these mitochondrial genomes, let's focus on haplogroup C, or C1. So C1b, C, and D came into the Americas and spread throughout the Americas, and CA may have expanded back into East Asia. Right? This is a hypothesis that we have. So over this last year, some additional genetic data has come out, and we've also been running a few simulations that's causing us to rethink some aspects of this model. And so here's a paper that just came out in PLAS 1 this last year, and it shows that C1b, C, and D are in high frequency in the Americas, possibly as a result of expansion into the Americas, bringing these mitochondrial genomes into the Americas. But then you also see, and then there's C1a, which we hypothesize was a back migration out of Beringia back into Northeast Asia. But over the past year, we've been able to see a haplogroup C1e that you find in Iceland. And so this is a head scratcher. How did that get into Iceland if most of the C1 haplogroups were in the Americas at the time, or in low frequency in Northeast Asia? And then recently there was this C1f lineage that was identified in an ancient individual in Western Eurasia. So it seems like the C1 haplogroup may not have been a result of expansions into the Americas or out of Beringia into the Americas and then back into Northeast Asia. Maybe C1 could have evolved somewhere in Eurasia a little bit earlier on in time. This kind of made sense along with the simulations that we were running, because we saw that genetic variants or new mitochondrial genomes, derived mitochondrial genomes that we simulated to evolve were still in extremely low frequency after a new mutation happened. And so often when the founder effect of individuals moving into the Americas happened, we lost these mitochondrial genomes due to genetic drift. It was very difficult. Well, there's no simulations that we've run where we see the number of mitochondrial genomes in matching the model that we expected. Right? So there seems to be some modifications that we need to make, and perhaps maybe there was a smaller population in northern Eurasia, and there was some isolation, but the expansion into the Americas was extensive enough, and there weren't people there that you get to see these mitogenomes in high frequency, but you don't see them in high frequency in other parts of the world where they've either gone extinct or they're in extremely low frequency. Okay, so this is how much time do I have? One more minute. Okay, so this is the last thing that I wanted to discuss, and this is a principal components plot. And so this is from our paper that we published in PLOS Genetics last month, where we have worldwide genomic-wide data sets, as well as groups that we sampled in British Columbia, which are the black dots or figures, right? So what we see in this principal components plot is that these dots that are closer together are more closely related to one another. Right? And the farther they are, the more distantly they are related to one another. And so we see in this plot is that Northwest Native Americans kind of have a vector or a span where some of these individuals are closer to Europeans. And this is likely a result of European contact and European admixture, or intermarrying after contact. But what's unique is that there's a different strata for Northwest Native Americans compared to the Central and Southern Native Americans that have been sampled so far. And so this strata suggests that there's a differentiation between Northern Native Americans and Central and Southern Native Americans in the genomes of living individuals. And so one possibility is that this is a result of the serial founder effect model. Sorry about the typo here. There should be a effect here. And so the serial founder effect is a model that suggests that as populations expanded out of Africa to people the rest of the world, there were a series of founder effects. And a founder effect is when a small group of individuals that is unrepresentative of the genomic diversity moves and founds a new population, and then grows, and then there's another founder effect as another unrepresentative group splits off, moves, and then grows. And so the expectation of this model is that as you move out of Africa, you should see a decrease in genetic diversity. And you can see that there is roughly a decent correlation with decrease in genetic diversity. In the Americas, you can see that there's some deviation, probably because of European admixture. But you roughly see that Native North Americans have more diversity than what you would see in Central and South Americans. So this could be the explanation for this pattern, or it could be a result of additional movements into North America from Northeast Asia, where as these individuals moved into the Americas, they admixed with the individuals that were already there. I believe that's it. Thanks.
Thank you.